Hello students, in today's lecture we will be discussing the development of female gametophyte, which is the embryo sac. So this topic will be covered in three segments. First we'll talk about the structure of ovule, then we'll move to the process of Megasporogenesis, and then in the end I will be talking about the process of Megagametogenesis, which is the development of embryo sac. So first we'll talk about the structure of ovule. So ovule is present inside the ovary. After fertilization, the ovule is converted into the seed. So this is the well labeled diagram of the ovule. First we'll talk about placenta. This I have already told in my previous video also. So placenta is the tissue which connects your ovule to the ovary. Then we have the funicle. Funicle is basically a stalk-like region which connects your ovule to the placenta. It's a stalk-like region connecting ovule to placenta. Then we have the helium. Helium is basically a junction which connects your funicle to the body of ovule. It's a junction that connects funicle to the body of ovule. Then we'll move to the inner part of the ovule. So first we'll talk about the integuments. So there are two layers. This is the outer layer and this is the inner layer of integuments. So integuments are basically the protective layers. Okay, its role is that they are protective layers. In most of the angiosperm, there are two integuments, an outer and an inner. So that is called as bitegmic. Okay, when there are two integuments present, it is called as bitegument. When the integument is absent, so that condition is called as ategmic, and the example is santalum. Okay, so here the integument is absent. Clear? Then we have the nucellus. This inner layer outside your embryo sac is the nucellus. So basically nucellus is a mass of parenchymatous tissue. It's a mass of parenchymatous tissue. If the nucellus, this complete wall of nucellus, if it is persist in the seed, we call them as perisperm. Okay. So after fertilization, I told you that the ovary is converted into the fruit and the ovule is converted into the seed. So if these integument layers, they finally make the outer walls of the seed, the hard seed coat. But if that nucellus, it still persists in the seed, we call him as perisperm. Okay. Then we have inside the embryo sac. This is the inside embryo sac, the female gametophyte, which has different structures. I'll tell you in detail later when we'll talk about the development of embryo sac. Here I just give you a little brief that these are called as the synergids. The two cells are called as the synergids. We have the egg cell, the two polar cells, polar nuclei, The complete cell is called as the polar cell, and we have 3 antipodals. So this, the role of individual cell, we'll read in detail when we'll talk about the development of embryo sac. So inside portion is your embryo sac. Then we have 2 important ends. One is the micropyle, another one is the chalaza. Okay. So micropyle, it is this tip region, this apex region is called as where you don't find integument is the micropyle. So this is the region from where the pollen tube enters. So basically this region doesn't has the integument, reason being because from here only the pollen tube it enters. Then the region which is just opposite to your micropyle is the chalaza. So this region is region opposite to micropyle is called as chalaza. So in this region the integument walls and the nucellus meet. Okay, so here you can see at the chalazal end the outer inner integuments and the nucellar cells they meet. So it is just opposite to the micropyle. Okay, so with this I have covered the structure of ovule. Let's revise once again. We have the placenta which connects the ovary to the ovule. We have the funicle which is a stalk-like region which connects the ovule to the placenta. Then we have the helium. It is the junction that connects the funicle to the body of ovule. Then there is nucellus, which is a mass of parenchymatous tissue, and if it persists in the seed, it is called as perisperm. Inside the nucellus, we have the embryo sac, okay, which has the egg cells, synergids, polar nuclei, and antipodals. Then we have 2 protective layers of integument. So integuments in angiosperms are generally bitegmic. If it is absent, it is called as ategmic. The example is santanum. Then we have 2 ends. One is the micropyle, the tip region where the integument is absent because this is the region from where the pollen tube enters. The region which is just opposite to the micropyle is the chalazal, and here the integument walls and the nucellus meet. Okay. So now we will be talking about the different types of ovule based on its orientation. Okay. So now we'll talk about the different type of ovules. So based on the orientation of ovule, we have different types. We have 6 different types, and I will explain each of the types. So first we'll talk about the orthotropous ovule. So let's revise once again. So this is the micropylar end, the opposite one is the chalazal. Then we have, this is the funicle, this is your micropyle, and this end opposite to is the chalazal. Okay, here is your egg cell. So in orthotropous, the micropyle, chalaza and funicle, they all lie in the same line. Okay, you can see micropyle, chalaza and funicle, they all lie in the same line. So this is called as orthotropous ovule. Then we'll move to the hemianatropous. Hemianatropous means when the ovule takes a 90-degree angle. So here, this is your funicle. So here your funicle is at right angle with your integuments and nucellus. So these 2 layers are of integuments, and the inside one you have the nucellus. So when the funicle is at right angle with the nucellus and integument, it is called as hemianatropous. Okay. Then compylotropous ovule. Compylotropous ovule is when the ovule is curved. When the ovule is curved, it is called as Campylotropous. Simple. Then another type we have is Amphitropous. So in Amphitropous also the ovule is curved, but the difference with Campylotropous is that in Campylotropous the ovule curvature is moderate. Okay, the curvature is moderate curvature. But in Amphitropous, this curvature, this ovule which is curved is so deep is major that it changes the shape of the inside embryo sac. So here if you can see, the embryo sac is horseshoe shaped. Here the embryo sac is horseshoe shaped. Okay, so the difference between Amphitropous and Campylotropous is that in Amphitropous the curvature is very strong, because of which the inside embryo sac is horseshoe shaped. But in Campylotropous the ovule is moderately curved. It doesn't changes the structure of the inside embryo sac. Okay, then we move to Anatropous. The Anatropous is the most common. It is the most common ovule orientation which is found in angiosperm. Okay, here your ovule is turned at an 180-degree orientation, because of which your micropyle and your funiculus, so this is your micropyle and this is your funicle. So because of this 180-degree turn, the micropyle comes in close proximity with the funicle. So that is Anatropous, the most common one. Then we have the Cercinotropous. So in Cercinotropous, the ovule takes 360-degree turn. So because of which, if you can see, the funicle is wrapped around the ovule. So because of this turn of 360-degree, the length of funicle increases so that it covers the entire ovule. So here the length of funicle increases to cover the ovule. Okay, so let's revise once again. We have 6 different types of ovules based on its orientation. First one is Orthotropous. Orthotropous, when the micropyle, chalaza and funicle, they all lie at the same line. Hemianatropous, when the funicle is at right angle with nucellus and integuments. Campylotropous, when the ovule is curved but moderately. Amphitropous, when the ovule is curved so strongly that it changes the structure of the embryo sac and it forms a horseshoe shaped structure. Anatropous, the most common one found in the angiosperm. Here the ovule it takes a turn of 180-degree so that the micropyle comes in close proximity with the funicle. Then we have the Cercinotropous, where the ovule takes a turn of 360-degree, and because of that the length of funicle it increases to cover the entire ovule. Okay. So now we will talk about the process of megasporogenesis. So like I said, megasporogenesis is the formation of megaspore from megaspore mother cell by meiosis. So how the megaspore is formed from megaspore mother cell? So like I explained in microsporogenesis, there is an archisporal cell involved. So again here also an archisporal cell is an hypodermal cell. So an archisporal cell which is diploid. So it's an hypodermal cell from the nucellus which gets enlarged. It has a dense cytoplasm and a prominent nucleus. It undergoes mitosis and it forms the primary parietal cell and primary sporogenous cell. Okay. Now the fate of both the cells are pre-decided. The primary parietal cells, it contributes to the nucellus wall around the megaspore mother cell, and the primary sporogenous cells. So few of the cells of primary sporogenous cell, it differentiates, it undergoes mitosis and it forms the megaspore mother cell. Okay, so megaspore mother cell is also diploid. So till here all the division, everything is through mitosis. So the megaspore mother cell is formed. So megaspore mother cell also has a callose wall around it. Okay, now this megaspore mother cell, it undergoes meiosis and it forms megaspores. So there are 4 megaspores are formed. They have a callose wall around it. So they are arranged as a linear tetrad. So generally the arrangement is linear tetrad. Okay, till here everything is understood, right? That from an archisporal cell undergoes mitosis, forms primary parietal and primary sporogenous cell. Primary parietal forms the nucellus wall around the megaspore mother cell, and the primary sporogenous cell, some of the cells undergo mitosis and forms the megaspore mother cell, which has a callose wall around it. It undergoes meiosis and forms the megaspore. Okay, now this megaspore, so there are 4 megaspores, they are formed. So in flowers, what happens, out of these 4, the 3 degenerate and there is one functional megaspore left, which forms the embryo sac. This is very important. Understand that megaspores, there are 4 megaspores are formed, out of which the 3 degenerates, and it is only the one functional megaspore which forms the embryo sac. This functional megaspore will be haploid and it will form the embryo sac. Such a kind of development is called as monosporic because there is a single functional megaspore which is going to form the embryo sac, and it is also called as the polygonum type of development. Okay, so remember these two terms. A single functional megaspore going to form the embryo sac called as monosporic development, and it's the polygonum type. Polygonum type basically means that this embryo sac from the single functional megaspore, there will, when it will form an embryo sac, it will be 7 celled and 8 nuclei. This I will explain you in the next topic. Okay, so now we'll move to the process of megagametogenesis. That is how this megaspore forms the embryo sac. Okay, so now we'll talk about the process of megagametogenesis, that is the formation of embryo sac from the functional megaspore. The formation of embryo sac from megaspore. So as I have explained earlier that the development of embryo sac is monosporic. That is, when the megaspore mother cell divides through meiosis, it forms 4 megaspores, out of which 3 degenerates and 1 is the functional megaspore which forms the embryo sac. So the development we call it as monosporic. Because from a single megaspore the embryo sac is going to form. Now this is the megaspore. So now the megaspore divides through mitosis. Okay, I am writing as the first, as there will be 3 rounds of mitosis. So after the first round of mitosis, 2 nuclei are formed, and both the nuclei, they move to the opposite poles to later create a differentiation in the polarity. So one moves to the micropylar end, okay, and the another one moves to the chalazal end. Okay, now there is another round of mitosis and 2 nuclei are formed. Then there is third round of mitosis and 4 nuclei are formed at each of the poles, at the micropyle and at the chalazal. Now these nuclei, they moves to the, so out of 4 at the micropyle, the 3 remains at the micropylar end and the 1 moves towards the center. Likewise at the chalazal end also, the 3 nuclei, they remain at the chalazal and 1 moves to the polar nuclei. Okay, now there is the process of cellularization take place, that is the formation of cell wall around the nuclei. So when that has happened, this is the final embryo sac that you see. This is the embryo sac. So now what are each of the cells called? So the 2 cells at the micropylar end, these 2 cells, they are called as the synergids. And in the synergids, these finger-like projections, they are called as filiform apparatus. Now the role of synergids and filiform apparatus is that it guides the pollen tube. Pollen tube entry inside the embryo sac so that it can fertilize the egg. Okay, so it guides the pollen tube for its entry. So basically, when you will later study the process of fertilization, you will get to know that synergids, it releases some chemical attractants through which the pollen tube gets attracted. It enters through the micropylar end. Okay, so this is the role of synergid, that it guides the pollen tube for its entry. Then this is your egg cell. Okay, now this egg cell, I told you that in 60% of the angiosperm, the pollen grains, they are shed at the 2 cell stage. So before here, the generative cell, it divides and form 2 male gametes. One of the male gamete, it fuses with the egg cell. One of the male gamete, it fuses with the egg cell and it forms the zygote. Okay, so this is your egg cell. Now one of the nuclei moves to the center, another from the chalazal move to the, one of the nuclei from the chalazal and moves to the center. These 2 nuclei at the center, they are called as the polar nuclei. Okay, these two nuclei later fuses and they form the secondary nucleus. Okay, now what happens, because there are two male gametes, one of the male gamete fuses with the secondary nucleus. So I will write here, it fuses with one of the male gamete, and after fusion it forms the primary endosperm nuclei, which is triploid. You will understand it later also when we will talk about the process of fertilization. Okay, so one of the male gamete fuses with the secondary nucleus to form the primary endosperm nuclei, which is triploid. Okay, and we have the three cells at the chalazal end. They are called as antipodals. They are called as antipodals. Their role is that they are nutritive. They provide nutrition to the developing embryo sac, and they after that they degenerate. So they are temporary, they degenerate at the later stages. So we'll revise once again. This megaspore, which is haploid, okay, the one single megaspore, it undergoes 3 rounds of mitosis to form an 8-celled stage, to form 8 nuclei, to form 8 nuclei, okay, to form 8 nuclei. Then there is a process of cellularization where an complete embryo sac is formed, which has 2 synergids and 1 egg cell at the micropylar end, 2 polar nuclei at the center. This is also called as the polar cell. So 2 polar nuclei are in the center, polar cell, and 3 cells at the chalaza, okay, called as the antipodals. Now such a kind of development, it is called as polygonum type of embryo sac development. Why? What is the meaning of polygonum? That is, the development is called as 7-celled 8 nuclei. Okay. So after 3 rounds of mitosis, there is 8 nuclei are formed. 1, 2, 3, 4, 5, 6, 7 and 8. But if you count the number of cells, it is only 7. 1, 2, 3, 4, 5, 6, 7. So 3 at the micropyle, 3 at the chalazal, and a single polar cell in the center. So that is why it is called as polygonum, that is 7-celled 8 nuclei. Understood? So this is the whole process of megagametogenesis, that is the formation of embryo sac from the megaspore. So in the next lecture, I will be talking about the process of pollination. I will emphasize that if you are somebody who is preparing for the NEET exam or going for the class 12 both, I really want you all to subscribe to my channel, because I will be following the NCERT line by line. If you really like my video, please like, share and subscribe. Thank you.